major diseases by age. So as you can see, the incidence of the diseases increases abruptly after about age 50. Similarly, the healthcare cost also increases abruptly after about age 50. So healthcare cost is a surrogate marker for the incidence of all human diseases, because if we get diseases, we have to go to hospital. So it costs some money. So how about infectious diseases such as COVID-19? So of course, the number of COVID-19 patients are much higher in a younger age than in older age. But lethality of the COVID-19 patient also abruptly increase after age 50. So that means age 50 is a very important timing for the humans. Most of us feel the aging around age 50. Recently, research on aging in organisms other than humans have been actively investigated. So although it is very difficult to find a common standard of the aging across species, but mortality could be a good indicator. Actually, in the humans, mortality abruptly increases after age 50, and the increase in the mortality reflects the promotion of the aging process. So mortality can easily analyze. Then there are many organisms that show the increase in the mortality with age, such as humans, mouse, killer whale, and guppy. Importantly, these organisms show the typical aging phenotype. Surprisingly, there are many organisms they don't show the increase in the mortality with age, such as tortoise, lizard, naked mole rats, and crocodile. So as I told you, the increase in the mortality rate reflects the promotion of the aging process. So that means that these organisms don't show the aging phenotype? So the answer is yes. Recent two science papers clearly demonstrate that when tortoises were fed under the optimized condition, they show very slow or negligible senescence. So that suggests aging is a phenotype that can be avoided. Interestingly, although the elephant shows some typical aging phenotypes, such as change in the shape of the skull, but elephants usually don't get age-related diseases such as cancer. So this is a cover picture of the Newsweek. They say elephants don't get cancer. So we now know the reason why. Elephants don't accumulate senescent cells. I will let you know later. More interestingly, there are some immortal organisms. So Hydra is immortal. Do you know the Hydra? Hydra is a kind of jellyfish. You may see the Hydra in aquarium or so. Hydra can survive at least for 14 centuries with a constant mortality. So now the researchers investigating why the Hydra is immortal using the single-cell transcriptomic analysis. Then the research on aging using identical twins have suggested that aging process is regulated by only 20% of genetics and 80% of epigenetics. Epigenetics means environmental factors. These are the identical twins, but looks very different. She's a little bit more younger than her because they were raised in a different environment. So now we understand that aging can be regulated by the environmental factors. So I would like to tell you about organismal aging regulated by senescent cells. I told you the senescent cells before. And recently, tissue chronic microinflammation has been proposed as a major cause of aging. So accumulation of inflammation-inducing cells, such as the senescent cells, cause excess tissue chronic microinflammation. This chronic microinflammation likely decline various organ and tissue function, promoting aging process and enhance the onset of various age-related diseases such as cancers. So inflammation-inducing cells are composed of both immune cells and non-immune cells. Immune cells such as macrophages and T cells are necessary for the normal immune responses. Therefore, targeting immune cells appear to be very difficult. So in contrast, non-immune cells such as senescent cells accumulate with age and do not necessarily to be essential for the normal organ and tissue function. Therefore, targeting inflammation-inducing non-immune cells, namely elimination of them or suppression of their inflammatory phenotype, likely suppress the chronic inflammation, improving the age-related dysfunction, and suppression of the onset of various age-related diseases. So here I'd like to introduce you a little bit about cellular senescence. Cellular senescence was first reported by Dr. Leonard Heflick, a US cell biologist, almost 70 years ago. Unfortunately, he passed away last summer. He found normal human cells possess a finite number of population doublings. After cells have passed the limited number of population doublings, cells undergo senescence. So senescent cells can survive for a long time and secreted various pro-inflammatory cytokines and growth factors causing the tissue chronic microinflammation. So in contrast, most cancer cells don't show the limited number of population doublings, namely immortal. So recently, cellular senescence has been proposed to be induced by various genotoxic stresses such as terminal dysfunction. Terminal dysfunction means a limited number of population doublings because each cell division, terminal length becomes shortened. Cellular senescence is also induced by the oxidative stress, DNA damage, oncogene activation, or epigenomic abnormalities. Senescent cells show the various unique characteristics such as permanent cell cycle arrest, apoptosis resistance, epigenetic changes, and the secretion of the various pro-inflammatory cytokines growth factors, so-called senescence-associated secretory phenotype, SASP. SASP can cause tissue chronic microinflammation. Then we would like to remove the senescent cell in old organisms. So we found GLS1 is essential for the senescence, but not normal cell survival. GLS1 is a rate-limiting enzyme, the glutaminolysis, a glutamine metabolic pathway. Glutamine is the most abundant circulating amino acid. GLS1 catalyzes conversion from glutamine to the glutamate. So this reaction is the deaminase reaction, producing ammonia as a byproduct. But at that time, we didn't know the physical role of ammonia at all. Glutamate is also the source of various important biomaterials such as alpha-ketoglutarate and non-essential amino acid and glutathione. Glutathione is a major antioxidant in the cells. Alpha-ketoglutarate is the intermediate substrate of the TCA cycle. That's the reason why glutamate can produce ATP. So we would like to know which substrate is important for the senescent cell survival. So to make long story short, we finally found ammonia is very important for the senescent cell survival. Even in a normal condition, around 10% of newly synthesized protein form the misfolding. Usually misfolded protein were translocated to the lysosome for the degradation to amino acid. Then the amino acid can be reused for the new protein synthesis. So we found senescent cell shows excess protein synthesis. So excess protein synthesis means more production of the misfolded protein. So accumulation of the excess misfolded protein in the lysosome over the capacity, misfolded protein likely form the protein aggregates. These protein aggregates cause the lysosome membrane damage, leaking the intralysosomal proton ion to the cytosol, leading to the intracellular acidosis. So intracellular acidosis usually rapidly induces apoptosis in the cell death, through the mitochondria and PTP pathway. But senescent cells produce high level of ammonia through increased expression of the GLS1. So this ammonia can neutralize intracellular acidosis. Then senescent cells can survive. So we did not detect any lysosomal membrane damage or protein aggregates in the normal cells. So that means a transient inhibition of GLS1 selectively induces cell death in the senescent cells. So based on this concept, we examined whether the administration of the GLS1 inhibitor to the aged mice could improve a certain age-related dysfunction. Actually, we found the GLS1 inhibitor improved age-related kidney dysfunction, age-related to liver dysfunction, age-related to lung fibrosis, sarcopenia, and certain type of lifestyle diseases such as atherosclerosis or diabetes. Then we would like to know why senescent cells accumulate with age. So second topic is the immune surveillance of the senescent cells. So previously, certain type of senescent cell can be eliminated by host immune surveillance system. For example, upon CCL4-induced liver fibrosis, senescent hepatic steroid cells can be eliminated by NK cells. Upon oncogene activation, senescent hepatocytes are eliminated by CD4 positive T cells. So these observations cast the question that, are there any common mechanism to remove senescent cells during natural aging process? How can senescent cells escape from immune attacks and accumulate during age? So to address this important question, we examine the properties of the senescent cells accumulate in aged organisms and found a part of senescent cells express PD-L1. PD-1 interaction suppress T-cell function, induce immune tolerance, and protect the body from tissue damage by suppressing excessive immune responses. So, as you remember, Professor Tasuku Honjo, Kyoto University, got a Nobel laureate by the discovery of this PD-1, PD-L1 immune checkpoint. So we found part of senescent cells expressed PD-L1. So this observation has suggested a promising working hypothesis regarding the immune escape and the senescent accumulation. So there are two distinct types of senescent cells. One is a PD-1 positive senescent cell, the other is a PD-1 negative senescent cells. So PD-1 negative senescent cells are eliminated by host immune surveillance system, whereas PD-1 positive senescent cells escape from immune attack and accumulate with age and secrete various pro-inflammatory cytokines, growth factor, causing a tissue chronic microinflammation. Actually, we found PD-1 positive senescent cells show more stronger inflammatory phenotype than PD-1 negative senescent cells. So that means PD-1 positive cells are more worse senescent cells because they cause a strong tissue chronic inflammation. So actually, the PD-1 positive senescent cell express a much higher level of cytokines and growth factors. Then this slide summarizes molecular mechanism underlying the escape from the immune surveillance of senescent cell by PD-1 signaling. So usually the CD8 positive T cell recognize senescent cell through the endogenous retroviral peptide on the MHC1 molecule. I didn't tell you this mechanism in detail, but I don't have so much time. But CD8 positive T cell, recognized CD8 positive T cell can be activated by pro-inflammatory cytokines and growth factors secreted from the senescent cell. Then activate the CD8 positive T cell, eliminate these senescent cells. In contrast, PD-1 for the PD-1 positive senescent cell, PD-1 interaction strongly suppress CD8 positive G cell function. Although the PD-1 positive senescent cells secrete much, much more pro-inflammatory cytokine and growth factor, but they fail to activate CD8 positive T cell. Then this PD-1 positive senescent cell accumulate with age. So based on this concept, we examine whether administration of the anti-PD-1 antibody. So anti-PD-1 antibody can be now available for the many, many human cancers. Then actually we found that anti-PD-1 antibody improved a certain age-related dysfunction. So this is the final slide. As I told you, the tissue chronic microinflammation is a fundamental pathological basis of the aging. This causes many, many age-related diseases. So senescence accumulation is a major cause of the chronic inflammation. So elimination of the senescent cell may suppress the chronic inflammation, extending healthy lifespan. So now we are trying to apply this technology to humans as soon as possible. So thank you very much for your attention.
Thank you very much, Professor Nakanishi. So this concludes the keynote lecture sessions and the opening ceremony of the 5th Chile-Japan Academic Forum. On behalf of General Chairs, I would like to thank all distinguished speakers and participants for joining us today. Thank you very much. So before moving to the welcome reception, we would like to now take a group photo. So please kindly ask all participants to come this central part of this room. So, and some local staff can guide, so please